Okay, so yeah, like I said, okay, so yeah, today I will talk about two main components. Okay, one is about HPLC, another about the DNA. Okay, and we do it one by one. Okay, first about HPLC. Okay, and so for HPLC. Okay, so yeah, if you have taken the CHS 435. Okay, so you should have some little bit idea. Okay, and normally for HPLC instrument setup. Okay, has a following component. Okay, so yeah, that's here the solvent reservoir. Normally you have kind of two or more kind of solvent. Okay, and then the solvent will pump here the into instrument by the pump. Okay, and then connect with the auto sampler. Also, actually you can also do the manual injection. Okay. And then the come to the most important part of HPLC, that's column. Okay, HPLC column. Okay, so the sample separate on the HPLC column. Okay, and then go through the system. Okay, one by one. Okay, so that can be detected by the detector. Okay, eventually goes to waste. Okay, so depends. Okay, so sometimes if you have to run the preparative HPLC. Okay, so you collect the fragment. Okay. Yeah, so HPLC stands for a high performance liquid chromatography. Sometimes people also refer to it as a high pressure liquid chromatography. It's very powerful tool. Okay, to separate a mixture of the compounds. Okay, and has the following component. Okay, I already talked about it. Okay, right. And here's probably one video. Okay, share with you guys. Okay, so can save some of my words. Okay.
No.
No, give me one second. Let's see. Okay.
High-performance liquid chromatography,
But
could equally well stand for high-pressure liquid chromatography.
Okay, cool.
It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase non-polar. In paper chromatography, the solvent moves along the paper by capillary action. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high-pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, a component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a non-polar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
All right, okay, so that's here some video about here the general idea about here the HPLC. Okay, so overall, okay, so the most important is the separation of here the different components, okay, on the HPLC column. Okay, so pretty much a different here the substance, different here the compounds. Okay, our different here the polarity and the binding affinity to the stationary phase. Okay, so that's here the some kind of the packing material in the HPLC column. Okay, and so they have kind of a different ability, kind of absorb, kind of retention on the stationary phase. Okay, and then when you pass through the solution. Okay, so that's called the mobile phase. Okay, solvent. Okay, and then they have kind of different the distribution. Okay, so in the mobile phase and also the stationary phase. Okay, separation. Okay, and then absorption. Okay. And eventually, okay, so they will have kind of different the speed, okay, or kind of the travel distance, okay, along the HPLC column. Okay, by that way, okay, get the separations. Okay, and then just the continue the through the HPLC column by the mobile phase. Okay, and then you can separate individual component. Okay. So HPLC can be used here for the quantitative kind of also qualitative kind of analysis. Okay, so that's pretty useful instrument. Okay, so I will talk about that later. Okay. All right. Okay. So like I said, okay. So the most important is the HPLC separation. Okay. So based on the different migration. Okay. So between the stationary phase and also mobile phase. Okay. So stationary phase can just the packing material. Okay. So it depends on what kind of HPLC column and what kind of application. Okay. So can be C13, okay, or can be silicone, or even can be gel, or what other chiral material. Okay, so for example, I'm using the chiral HPLC column. Okay, and so for this CH454 DNA digestible project. Okay, so we use the C18 column. Okay. And for the mobile phase, okay, so yeah, just carry the sample, yeah, down through the HPLC column, okay, and so for the C18 column, okay, normally you have one organic the mobile phase, one inorganic mobile phase, okay. For our project, okay, we'll use the one TAA buffer, okay, so that's just the kind of aqueous phase, okay, and then another one is the nitride, okay, so that's the organic phase, okay. Right here, just showed you guys some kind of small cartoon, okay. So that's mixture, okay, pass through the HPLC column, okay, and then the separation, okay, and then the individual components, okay, pass through the detector, and then show on the as chromatograph, okay. So that's on the computer, okay. Right, so once the HPLC is done, okay, so the computer should generate some diagram, okay, called the chromatograph, okay. So that's give some kind of important information, okay. Two most important information. One is the retention time, okay. So that's the time from the injection until the P come out, okay. So if the HPLC condition set, okay, and also same components, same kind of enrollment rate, everything same, okay, and then for individual sample, okay, so it should come out kind of very kind of the set the time, okay. So time won't change, okay. So that's why you can use retention time to identify some kind of the sample, okay. So normally speaking, kind of people will run some kind of standard the HPLC, okay, and then get the retention time for individual the samples, okay, and then they will run unknown, okay. And so if it's the same conditions, okay, so if same component should come at the same return time, okay. By that case, so we can identify what component in the unknown samples, okay. Another important information is the area, okay. So that's used to quantify the samples, okay. So this the area, okay. So the how big of this peak, okay, so indicate how much of the sample, okay. So area actually kind of relate to the UV absorption of individual samples, okay. So that's the Beer lambda low, okay. So probably you guys already learned it, okay. So A, okay, so that's absorption, okay, so equals to the actually kind of is also the area size of the peak, okay, in the HPLC chromatogram, okay, equals to the molar absorptivity, okay. So normally I like call it kind of molar diffusion coefficient, okay, and the times kind of concentration times the cell kind of the path length, okay, and so normally speaking, kind of cell path length kind of set, okay. So for HPLC instrument, okay, and so if the molar absorptivity is also set, okay, so that you can directly use it to determine the concentration of the sample, okay. And here's just the one UV the chromatogram of the chlorophyll, okay, so 2, okay, so and so you can see kind of for this compound has the 2 peaks, okay, so and then you can label it lambda max and also the individual the molar integration coefficient, okay. And so for our HPLC analysis, okay, so we set the detector wavelength at the 260s, okay, because that's the best for all the 4 ATCG the base, okay. All right, so now come to problem, okay. So because they're different here, the base have different here, more absorptivities, okay. So in this case here, we have to normalize, okay. So and so that here the just kind of relative ratio of here the molar absorptivities, okay, and that's we call cofactors, okay. And one of the most important kind of here the work for my units is here the distinguish figure out here what's here the cofactors for for nucleoside, okay, ATCG, okay. So that's I will talk about more later, okay. All right. So HPLC pretty useful, okay. So can be using quite a variety kind of fields, okay. So like here the pharmaceutical, okay. So people use HPLC try to identify the counterfeit the drug products, okay, or maybe kind of determine the lifetime of the pharmaceutical product, okay. and also can be used for environmental analysis. Okay, so some kind of contamination in the water or some kind of the phenol. Okay, for example, phenol. Okay, in the drinking water. Okay, and also used in the forensics. Okay. So for example, identify some kind of stereo kind of in the urine or sweat or some even hair. Okay, so pretty powerful. Okay, because the HPLC has a very high sensitivity. Okay. And clinical, okay, for example, analysis of the DEET, okay, DET, okay, in the human urine. Okay. And flavor and also food. Okay, so most time it's just used for the quality control. Okay. And so for example, analyze the sugar amount, okay, in the fruit, juices. Okay, so and so if you pretty good at HPLC, okay, so it should be easy for you to find a job. Okay, I have several friends. Okay, so they only know how to run the HPLC, okay, and then you kind of find a pretty good job. Okay, so and here just kind of two pictures. Okay, one showed you the HPLC forest in the some kind of the pharmaceutical companies. Okay, so the whole floor. Okay, so the building. Okay, just take by all the HPLC. Okay, it's kind of the many HPLC. Okay, so and another one kind of the industrial HPLC. Okay, so far the HPLC we are running pretty much just the analytical HPLC. That's small kind of the scale. Okay. Sometimes kind of I may run some preparative HPLC. That's not so big. Okay. So for the industrial HPLC, okay, you can see that's the column. Okay, so huge. Okay, so that's we use a lot of solvent. Okay. Right. Okay, so that's about HPLC. Any questions? Right. So if you have questions, okay, so we can speak out or you can type in the chat window. Okay. All right. So next, I will talk about the DNA. Okay, and also nucleic acid. Okay, so. That's directly related to my units. Okay. Okay. So DNA is called the deoxyribonucleic acids. Okay. So the abbreviation kind of DNA. Okay. So this is one type of nucleic acid. Okay. And the basic units of DNA is called the nucleotide. Okay. Nucleotide. Keep in mind. Okay. And nucleotides has the three components. Okay, so phosphate and also the nuclear base. Okay, nuclear base. We will learn two kinds of nuclear base. One is the purine. Okay, one is called pyrimidine. Okay, and the last one is kind of the sugar. Okay, so either 2-priodeoxy ribose or ribose, regular ribose. Okay. Right. Okay, so we do it one by one. Okay, so first is the nucleobase. Okay, so nucleobase. Okay, so like I said, okay, two kinds of ones based on the pyrimidine, one's based on the pyrine. Okay, so three nucleobase based on pyrimidine. Okay, uracil, okay, thymine, and also the cytosine. Okay, and two nucleobase based on the pyrine. Okay, purine. Okay, so adenine. Okay, and also the guanine. Okay. Right, so you don't need to remember the number. Okay, so that's not required. Okay, and how to number the ring case structure, and for the pyrimidine case. So uracil only exists in the RNA. Okay, and then for the thymine only exists in the DNA. Okay, so that's one difference between the DNA and also RNA. Okay. Right. Okay, and then the sugar. Okay, so sugar. Okay, so actually this is the required case. So probably I need bring the notes here. Okay. Right, sugar. Okay, so that's normally the 5' membrane ones we call kind of the pentose. Okay. All right, and then for the pentose. Okay, so you need to learn how to the number it. Okay, because that's kind of important kind of determine the base structure and also the primary structure of your DNA. Okay. All right, so that's the sugar. Okay, so probably here. Okay. And when we number it, okay, so we start from this carbon. Okay, so so that's we call the primary. Okay, and then followed by secondary. Okay, kind of the 2' it's the 1' and then 2' and then 3' and 4' 5'. Okay. Please recall this one in your notebook. Okay. I may test even the unit exam. Okay. And also maybe unit quiz. Okay. So why we use prime? Okay, so reason for that here, the number actually taken by the base. Okay, so the base here, the number is not required, but the sugar number is required. Okay. Okay, so now we come to kind of second difference between the DNA and RNA. Okay, so for RNA case, so you have kind of the OH group under the second 2' kind of carbon. Okay, but for the DNA case, so this 2' kind of OH group become hydrogen. Okay, so okay, so that's why we call it kind of 2' dioxy. Okay, so it's kind of the OH group is gone. Okay, so and then ribose. And for the next key, we can talk about nucleoside. Okay, so because we are learning nuclear base and also the sugars. Okay, and nuclear base attached to sugar at the primary positions. Okay, the 1' position. Okay, so that's why I asked you guys kind of try to remember the case. And because this carbon. Okay, so probably still go here. Okay. Because of these carbons, the chiral carbon, okay, so and then when the nuclear base attach sugars, okay, so can go either from top or from bottom, okay, so you can have 2 ways, okay. And for the natural DNA, okay, also natural kind of RNA, okay, so we always attach from the top, okay, and then we generate a bond called the beta glycosidic bond. Okay, so that's from here. Okay, so beta. Okay, so alpha will be kind of from bottom. Okay, so sometimes kind of people make the nature kind of the DNA. Okay, so so they kind of get alpha 1. Okay, so sometimes can you see the base going down? Okay, so probably also. So that's actually kind of called alpha. Okay. But for the older natural the DNA, okay, so that's called the beta bond. Okay, so this one going up. Okay, so the beta bond kind of nuclear base going up. Okay, and then hydrogen going down. Okay, but for the alpha 1, okay, so alpha 1, okay, so hydrogen going up. Okay, nuclear base going down. Okay, so that's make sure you know how to draw it. Okay, so all right, okay, go back. Yeah, if you have any question, you can type in the chat window. Okay. Okay, and then now we can talk about the nucleotide. Okay, so that's actually kind of easy. Okay, just here the nucleoside plus phosphate. Okay, and the phosphate diester bond. Okay, and so the phosphate bond. Okay, so phosphate. Okay, so can attach either on the 3' position. Okay, or 2' position. Okay. Phosphate can go either here or get here. Okay. So doesn't matter. Okay, so 3' or 5'. Okay. Right, and then here's one example. Okay, so that's called 5' monophosphate canal. I don't think 5' mono the phosphate. Okay, so that means this the phosphate only one phosphate attached at the 5' carbon. Okay. Right. And so here's another example. Okay, so this one called uridine. Okay, 3' monophosphate. Okay, so that's phosphate attached at the 3' carbon. Okay. And not only can you attach one the phosphate, you can attach the 2 phosphate or 3 phosphate. That's called the diphosphate or triphosphate. Here's one example. That's actually the adenosine triphosphate. That's the abbreviations here. The ATP is one important the chemical store the energy. So for our body. And so this one case, so the 3 phosphate attached at the 5' carbons. Okay. Right. Okay. So here's just a little bit summary of your nuclear base, okay, nucleotide, and also your nucleoside. Okay. So and so just a little bit keep in mind. Okay. So your nuclear base name different from nucleoside. Okay. So as the name case, so for nuclear base. Okay. And then once we come back and talk about the nucleoside. Okay, that's become adenosine. Okay. So and then for the DNA case, so nucleoside case just as 2' deoxy before the nucleoside name. Okay. And pretty easy case, so that's nucleotide equal to nucleoside plus phosphate. And then the DNA units, that's the nucleotide, not nucleoside. So that's real DNA units. Right, okay, so now, yeah, once we understand the DNA units, okay, so now we can assemble the DNA, okay, so yeah, from a nucleotide, okay. So yeah, we first learn the primary structure of the DNA, okay, that's just the single strand DNA, okay. And so that's the DNA, yeah, just the based on its backbone. This DNA backbone, yeah, just has two components, phosphate and also the ribose, the sugars, okay. And so yeah, this phosphate, okay, so yeah, one end attached to one sugar of 5' and then another end, okay, so we probably use this one, okay. Another end attached to the next sugars, okay, at the 3', okay, so yeah. So in this case, here the 2', 3', okay, that's linked together, okay, and then you generate the DNA single strand, okay. And yeah, the DNA kind of the phosphate, okay, so yeah, normally we call it the phosphorus kind of diester, okay, phosphorus diester, okay. Okay, so yeah, quite often yeah, you see some people said kind of this DNA strand sequence, the AATT, CCGG, something like this, okay. So that's actually indicated the direction of the DNA from 5', okay. So that's kind of important, okay. So yeah, don't get confused, okay, because the DNA can either start from the 3' or 5', okay, and make sure that normally okay, common sense, okay, so that's the way indicate the DNA sequence that's from 5', okay. For example, this is one example, okay, so yeah. From 5', okay, so that's we go these directions, okay, so that's CATG, okay, so that's 5', CATG, okay. And quite often, people even ignore the 5', okay, so that's you have to use a sense, okay. So that means this DNA strand actually indicated the start from 5', okay. So otherwise, they get confused, okay. So yeah. Questions? No, all right. Yeah, from 5' end to the 3' end. Okay, 5' okay, so that means the start from 5' carbons, okay, and then from one the DNA nucleotide, okay, and then go next one until the 3' of the end of the final nucleotide. Okay. Right, so that's the primary structure, and then we come to the secondary structure. Okay, secondary structure is the order how to arrange the DNA strands. Okay, so yeah, normally speaking, the DNA strand in a duplex secondary double helix the DNA the secondary structures. Okay, so there are also some other unusual the secondary structure like maybe the quadruplex. Okay, so but that's not common. Okay, for the double DNA helix strand case, so that's just the way case so DNA case so strand in the anti-parallel kind of the strand coil kind of in the right manners, okay, and then in the around the same axis, okay, and so the most famous the DNA double helix case that's B form the DNA case, so that's the proposed by the James Watson and also the Frank kind of Crick case, so that's the more famous kind of founding case. Right, and then for the DNA double helix case, that's assembled through the DNA base pair, okay. So that's probably you already know that kind of adenine to the T thymine, okay. So for the AT base pair, okay, so they can build up one actually kind of two hydrogen bond, okay. And another base pair is the CT base pair, okay. So guanine and also the cytosine, okay. So that's the base pair can form 3 hydrogen bond, okay. So that's much more strong, okay. So and so for AT, okay, only about 10 kcal per mole, okay. But for the CG, okay, base pair, okay, 17 kcal per mole, okay. So that means if we are in one DNA action of the double helix case, so if there are more the CG pair, okay, so that means the more stability of your DNA kind of the double helix case. Right, okay, so if all the base compared to each other, okay, so that's for the 2 DNA strand, okay, then we can form the DNA double helix case. Right, okay, so just keep in mind, okay, so the DNA double helix in the anti-parallel case, so that means one strand, okay, so start from 5' to 3', okay. Another strand start from the opposite directions, okay, so actually kind of from 3' to 5', okay. And so in this case, when you talk about kind of complementary strand for another DNA. Okay, so yeah, make sure you understand it's from 5' or from 3'. Okay, that's quite a common mistake. Okay, so I remember. Okay, so yeah, when I was a grad student. Okay, so yeah. Another postdoc okay, try to order some the DNA strand for his kind of the complementary strand for his kind of DNA strand sample. Okay, and then he just kind of okay, so for example his DNA strand. Okay, so it's kind of a kind of CG. Okay, all right. Okay, so that's but when you order kind of complementary strand. Okay, so keep in mind. Okay, so that's from 5'. Okay. Okay, and then complementary strand. Okay, so yeah, should do this way. Okay, so that's CGTT. Okay, and then reality. Okay, so that is complementary. Okay, and when he ordered one. Okay, so he ordered kind of a totally opposite one. Okay, so that make sure you understand this. Okay, so don't make a mistake. Maybe I will in the quiz I'll test you kind of what's complementary for this one. Make sure you give me the right answer. Okay. All right, so there are several models can show the DNA double helix. Okay, so and the ones just kind of fully kind of extend out. Okay, so show you kind of anti-parallel kind of the strand orientation scheme. And another one just uses stick. Okay, so this stick model actually kind of pretty useful. Okay, so clearly showed you the geometry and the dimension of your DNA strand. Okay, so some kind of number please keep in mind. Okay, so for the 1 full turn of DNA. Okay, so 1 full turn. Okay, so that's about 3.4 nanometer. Okay, also 34 angstrom. Okay. And then the DNA base pair distance. Okay, so that's about 0.34. Okay, so nanometer, 3.4 nanometer, 3.4 angstrom. Okay. So that means the 1 turn of DNA is about 10 base pair. Okay, around 10 base pair. Okay, that's the meaning for the B form DNA. Okay, so and then for the B form DNA, okay, you can clearly see kind of some there's a major growth. Okay, so sometimes when enzyme binds to DNA. Okay, so can bind through the major growth. Okay, and also kind of minor growth. Right, here's the space fading model. Okay, so that's more close to DNA real structures. Okay, and so that's kind of very beautiful kind of DNA double helix. Okay, right, okay. And so, like I said, okay, so the major the DNA secondary structure is the B form DNA. Okay, but there are also some other double helix. Okay, so B form DNA. Okay, so the just 20 angstrom thick and then 34 angstrom kind of per 10. Okay, so 10 base pair. Okay, and but for A form DNA. Okay, so it's also right-handed. Okay, but it's much thicker, but also the shorter case than the B form DNA. Okay, so only the 29 angstrom per 10 base pair. Okay, and very interesting. There's also another DNA, the double helix. Okay, so that's called a Z form DNA. Okay, so these strands actually kind of left-handed. Okay, so different from A form DNA and also B form DNA. Okay. Right, so here's some models for the A form DNA, B form DNA, and also Z form DNA. Okay, so you can see kind of for A form DNA. Okay, so that's much thicker but also shorter. Okay, for B form DNA. Okay, so that's kind of the narrow. Okay, and also the longer. Okay, and the Z form DNA. Okay, quite unique. Kind of left-handed. Okay. So that's about the DNA secondary structure. Okay, but in our cell. Okay, so the DNA packed in kind of the very dense structure. Okay, and that's come to kind of the tertiary structure of DNA. Okay, so that's 3-dimensional arrangement of the nucleic acid. Okay, so normally it just refers that kind of supercoiling. Okay. And so there are several types of the tertiary DNA structures. Okay, so one's the easy one, simple ones, the circular DNA. Okay, so it's kind of DNA double helix in the ring structures. Okay, and so to pack the DNA into the tertiary structure. Okay, so some enzyme. Okay, or some kind of the other biomolecule will come in kind of play important role. Okay, so for example some histone. Okay, will come in. Okay, help to pack DNA into the chromatin. Okay, So here just show some kind of some information. So that's circular DNA, and then there's some other super coil of DNA. And so normal DNA, so surrounded about the histone, and then pack kind of more tighter structure called nucleosome. And then some other enzyme further pack the nucleosome into the highly packed structure, and eventually form the chromosome, and so that can be fit into the nucleus of our cell, So here's a more interesting video, show the whole.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
Yeah, that's pretty nice video, showed you how the DNA pack into the chromosome, All right, so next, we'll talk about RNA, So RNA has the similar structures, and to DNA, but there are some kind of the difference, So showed here, So first, so the sugar units of RNA is the ribose, So there's the OH group at 2' carbons, And then second case, so the pyrimidine base of RNA is the uracil, and then the different format of the thymine case, so in the DNA. And so third difference is the DNA RNA case, so normally exists as single strand case. So DNA normally exists as kind of the double strand case. So that's 3 difference case. Okay, so RNA has kind of much more many more kind of other functions than the DNA. DNA normally just the storage of the gene information. So RNA so depends on its function. Has the following kind of several kind of types of RNA. The first one here, the ribosome kind of the RNA. So this is kind of important component here in the ribosome. Ribosome kind of the location to convert the RNA into the protein, so make the protein. And then next is the transfer RNA called also tRNA. So that's this the RNA can bring the individual nuclear amino acid onto the ribosome and then make the proteins. And the last one, the messenger RNA, so also called mRNA. So that's just carry the gene information from DNA to ribose, and then make the proteins. And so genes kind of just a section of the DNA that's coded for certain proteins, and each unique gene has the unique sequence of the base, And unique sequence basically we call it product of a unique protein, and eventually so this protein combination sometimes and generate the unique phenotype. So for example, you think that that's the dog is kind of long hair, this dog kind of short hair, so that's this dog kind of black, and another dog kind of yellow or white, so that's also called phenotype. That's actually all based on the different protein concentration. Right, and so for the synthesis of the DNA from the RNA from the DNA, okay, so mRNA from DNA, that's we call the transcription, okay. So we use the DNA as the template, okay, and the base pairing, okay, make the mRNA, okay. And so the base here arranged in the triplet, okay, so called codons, okay, okay. Codon, okay, so that's three nucleotide, okay, so nucleotide, okay, so coded one amino acid, okay, and in this case, based on the codons of the mRNA, so you can make the proteins, okay. So that's the genetically code, okay. So this one just get a little bit idea, okay. So individual codes, okay, code corresponding one kind of amino acid, okay. All right, so here's kind of central dogma, okay. So pretty much pretty much all the lives, okay. So use DNA as a template, okay. So and then DNA can replicate itself, okay. And if we use DNA to make the RNA, okay, that's called a transcription, okay. And then if you use RNA as template to make the protein, that's this process we call the translation, okay. Okay, so now we come back to the DNA, okay. So so for our project, okay, we need to use some enzyme to cleave the DNA, okay, and also try to generate useful information, okay. So enzymes very important here for manipulating DNA, okay. So there are several types of enzymes, okay. Here show you some, okay. So include the DNA polymerase, okay. So that's just to make the DNA, okay. And the kinase, okay, alkaline kind of phosphatase, okay. So that's so somehow kind of cleave off here the phosphate of the DNA, okay. And nucleus, okay, nucleus is just here the break up down the DNA, okay. So in special ways, okay. And then in that case, so turbo isomerase, so that's change the geometry, okay. So for example, the nucleosome, okay, pack into some kind of tight structure, okay. So that's those kind of enzyme will play the role, okay. And for our DNA, the digestion project, okay, will involve the two enzymes, okay. One's alkaline phosphate, okay, another nucleus, okay. We'll talk about this enzyme one by one, okay. And before that, okay, so we'll talk a little bit about the buffer and those solutions, okay, because that's kind of important for our project, okay. And so for each individual enzyme, okay, so they have only show their activity in certain buffer conditions, okay. That's why the buffer is kind of critical for the enzyme, okay. And so for ideal kind of buffer solution, okay, so normally around the pH pK around like the 6 to 8, okay, so that's the close to our body environment, okay, so and so should be kind of chemical inert, okay, so you don't want to kill the enzymes, okay. And also the bipolar, okay, so to be sure can dissolve in aqueous solution, okay, and then not membrane permeable, okay. No toxic, okay, so no inexpensive, okay. And also the salt and the temperature indifference, okay, so you don't want add some salt, okay, change greatly, okay, of the buffer kind of conditions, okay. And so for our the DNA digest, okay, so we'll use trace buffer, okay. All right, okay. The trace buffer full names, the trace, the hydroxy methyl amino methane, okay. So you are required to know the trace structures, okay. So actually kind of not hard, okay. So here. So if you took my organic chemistry, okay, so you should able to draw the structure of your trace based on its name, okay. So methane, okay, methane is kind of CH4, okay. So we normally when I teach the organic chemistry, okay, or taught student, draw the structure based on name, do the backward, okay. So we draw the kind of base structure of methane kind of CH4, okay. And then we do the amino, okay. So that means here one hydrogen replaced by the one hydrogen on the methane replaced by the amine group, okay. H2, okay, that's amino, okay. Right, and then we draw the kind of trace, hydroxyl methyl, okay. Hydroxyl methyl, okay, that's here CH2, okay, and then OH, okay, that's hydroxyl.
Can you slide the camera, Leo?
Oh, sorry. Right,
Thank you.
that's it. Yeah. Right. So that's here the hydroxyl methyl, okay. That's hydroxyl, okay. That's here the methyl, okay. So just here methyl group C3, okay, so you replace here the 1H which OH group, that's we call kind of hydroxyl methyl, okay, right? Trace hydroxyl methyl, okay, that means there are 3 these kind of units. Okay. C2 OH, right? So that's here the trace. Okay. Right, so make sure you recall the structure, okay. Right, so we are from trace itself, okay, you can see there's kind of amine, okay. Amine is the relatively the basic, okay. So that's why the trace pK is around 8, okay. And so yeah, a little bit the basic, okay. And so in this case, yeah, for the real trace buffer, okay, so you have to add a little bit the HCl, okay. So yeah, so that's why normally when you purchase trace, okay, so that's here the trace buffer is comes kind of say trace HCl, okay. So that's here yeah, neutralize here this here the amine, okay, and then you can adjust the pH to around 7, okay. Oh yeah, right. You guys get here the structure, okay. So that's trace buffer, okay. That's surprise, okay. So yeah, lot of the graduate even professor don't know the trace structures, okay. So make mistake. So, right, okay. Okay, and then yeah, now yeah, for the enzyme activity, Martin on trace. So I don't know what it means. So the all the trace, okay. So base structure is the methane, okay. So this is the methane, okay. Right, okay, and then yeah, methane will just replace the hydrogen, okay. So with the different starting group, okay. So yeah, one is amino, and then other 3 is the hydroxy methane, okay. So yeah, and if you refer to this methane, this is methyl, okay. So that's just here the branch connection, okay. So connection, okay. So that's here no special functions, okay, for the methane, okay. Right, yeah, that's right, okay. So if you have more questions, just type in the chat window, okay. Okay, and so yeah, but yeah, for the reality, okay, so yeah, if you wanted the enzyme to play the function in the reactions, okay, in the solution, okay, so only buffer is not enough, okay, so yeah, you have to add other components, okay, that's showed here, okay. So yeah, sometimes kind of salt, okay, so yeah, that's just here the try to keep a certain ion strength, okay, for the solutions, okay. So normally kind of sodium chloride, okay, or potassium, the chloride, okay, something like this, okay, phosphate, okay. And for some enzyme, okay, so yeah, if you want to the turn the function of the enzyme, okay, so you have to add some kind of divalent the metals, okay, like the magnesium, okay. So for example, yeah, for our DNA digestion project, okay, so we have to add the magnesium chloride, okay, so turn on the activity of the enzyme, okay. And also you add some other function, component, okay, so like glycerol, okay, so that's try to stabilize the enzyme structure, okay. And EDTA, okay, try to chelate the divalent kind of cations, okay, try to also kind of important kind of storage of the enzyme solutions, okay. And also sometimes the people add the beta the capital cellularic the ethanol can also kind of the cell threot, okay. So that's here because in the protein also enzyme, okay, so has some the sulfur bond, okay, to the ICH, okay. So yeah, you add those components, okay, try to stabilize this the ICH, okay, so we prevent the formation of the disulfide bond, okay. And also some kind of other the proteins, okay, also some other cofactors, okay. So and for our DNA digest, okay, so in addition to trace buffer, we will also add the magnesium chloride, okay, solutions, okay. Okay, and for the nucleus, okay, so that's where we'll talk about the enzyme one by one, okay. So yeah, the one enzyme we use in the DNA digestion project is the SVP, okay, and that's belongs to a nucleus, okay. So nucleus just cleave off here the DNA, okay. And there are 2 big categories for nucleus. One's called the exonucleus, okay. That's just a chop off the DNA from end of the DNA molecule, okay. So yeah, just one by one, maybe some kind of one more time, okay. Right, another called kind of endonuclease case. That's here break up here the DNA from inside of here the DNA structure case. And there's one special the endonuclease called the restriction the enzyme case. That's one very special case. Only cleave off the DNA at a certain position and based on certain DNA sequence. Okay, I'll talk about that later. Okay. Right, we so first do the exonuclease. Okay, so there are two example for the exonuclease. One is called exonuclease 3. Okay, so that's this one can cleave with double strand DNA. Okay, and then direction from 3' to 5'. Okay. Another example is here the exonuclease 1. Okay, so this one also cleave with the DNA from 3' to 5'. Okay. And it's just here from one end to another, okay, but only work on the single strand DNA. Okay, more useful to remove some the unextended primer. Okay, so just here you want to make here the DNA strand evenly. Okay, so double helix evenly. Okay. Right, and then some example for the endonuclease. One is here the Dynease 1. Okay, so this strand kind of quite interesting. Okay, so can cleave both double strand DNA, also the DNA strand, single strand DNA. Okay, and so randomly, okay, anywhere. Okay, and if you want to cleave the double strand DNA, okay, you need to add a magnesium kind of salt. Okay, if you want to cleave the single strand, okay, you use magnesium salt. Okay. And so, but if DNA bound with some the enzyme, okay, and then Dynease 1 kind of enzyme cannot cleave the bounded part. Okay, so that's why it's very useful to define the binding site of the DNA with the proteins. Okay, so I'll give some example. Okay, and restriction enzyme. Okay, that's we'll talk about the later. Okay. So here's one example case. So use the Dynease 1 case to try to figure out where the protein bound to double helix DNA. So first, okay, so you need use the P32 label DNA strand. Okay, so in this case, you can use the gel to visualize the DNA strand. Okay, so right. So if there's no protein bounded to double helix of the DNA, so then Dynease 1 can cleave off DNA double helix anywhere. Okay, so in this case when you run some electrophoresis gel. Okay, so you see kind of all the kind of component. Okay, for the DNA sequence. Okay, so but here if some protein bound to the DNA double helix. Okay, and then when the DNA come to Dynease 1 cleave the DNA cannot cleave this bounded part. Okay, and then just cleave the exposed part. Okay, and then there's some kind of blank part. Okay, showed on the electrophoresis gel. Okay, so that's here you can figure out where is the protein bound to the DNA strand. Okay, so pretty interesting. Okay. Right, okay, so now just some example for the restriction enzyme. Okay, so like I said, okay, restriction enzyme can only recognize a certain DNA sequence, okay, and only can cleave off certain DNA sequence positions. Okay, so for example, here's kind of actually I use the BAM-H1 okay, before. Okay, so only recognize this DNA sequence, and then actually when this enzyme cleave off DNA double helix, okay, not evenly cleave off the CGG base pair. Okay, so for one DNA strand, and then GG base pair. Okay, from another DNA. Okay, so not so even. Okay, very interesting, very useful. Okay, so right. Okay, so for the RDA digestion project. Okay, so the first enzyme we use called SVP. Okay, that's actually belongs to nucleus. Okay, and so this one is just the endonuclease. Okay, cleave off DNA just everywhere. Okay, so and so it's belong to the phosphorodiester. Okay, so let's break up the phosphorodiester bond. Okay. Once it come in, okay, so just break up DNA into the individual nucleotide. Okay, so nucleotide. Okay, and but the nucleotide is 2-polar for HPLC. Okay, so in this case, we have to apply another enzyme. Okay, called the alkaline phosphatase. Okay, so that's the cleave off the phosphate. Okay, of the nucleotide. Okay. Right, just remove 3' or 5' phosphate. Okay, so from the nucleotide. Okay, And so in this lab, okay, so we use the enzyme called the BAP, okay, bacterial alkaline phosphatase, okay. So that's only converted the nucleotide to nucleoside. Okay. The nucleoside kind of much less polar, okay, because the phosphate kind of too polar, okay, not easy to control. Okay. And the nucleoside is much less polar, okay, and then that can be analyzed by the HPLC, okay. Right. So yeah, here's kind of give you one example of what's going on. Okay, so yeah, for the organic digestion product. Okay, so we have kind of DNA sequence. Okay, and then yeah, we use both BAP and also ICVP. Okay, so yeah, cleave off the DNA strand into an individual nucleoside. Okay. And then we run the HPLC case. So yeah, and then because they're different here, the nucleoside has the different here the absorptivity on the DNA column. Okay, and then they will have a different here the retention time. Okay, and then show kind of different signal on the chromatograph. Okay, and then eventually, okay, so we can based on the nucleoside kind of retention time. Okay, identify what kind of nucleoside in this here the DNA strand. Okay. And also based on the integration, okay, so integration kind of size of the PK, so the intensity of the PK, and figure out how many ATCG in individual the DNA strand. Okay. So that's we will do it in this project. Okay. All right. Okay. So here's just another example. Okay. So like I said, okay. So we use the SVP to cleave off the DNA, okay, and then use the BP cleave off the phosphate. Okay. So regular DNA, okay, so after we apply these two strands, okay, all convert kind of SVP, okay, all convert into the nucleoside, okay. But if you do some modification, okay, so so that's one project I did when I was the PhD student, okay. So I linked the actual kind of 2' position, okay. So remember the phosphate linked through kind of 3' and also the 5' positions of the nuclear tide, okay. But here, if I link here the DNA through the 2' positions, okay, which are kind of double the backbone, okay. And then if we apply the DNA the enzyme, okay, so that's can only cleave off here the 3' 5' the phosphate backbone, but cannot do anything to the this the man-made backbone. Okay. So in this case we can detect this the special we call the oligo the nucleoside here the structures. Okay, so that's some kind of HPLC analysis. Okay, so we have some the DNA modified part of the DNA strand by this the this the man-made the backbone. Okay, and then this man-made backbone cannot be break down by the enzyme. Okay, so that can be detected by the HPLC. Okay, so that's here's some kind of we call it U2, okay, so it's kind of 2 nucleoside, okay, and then U 3 kind of 3 nucleoside, okay, connect together, okay, and then U5. That can be detected even by the HPLC, okay. And here kind of another example, okay, so even eventually we can add the link here up to 8 the nucleoside together, okay, and then through this the linker, okay, and then that can be detected by HPLC, okay, pretty unique. Okay. And then unlinked part, okay, still can be cleaved off by the enzyme, okay, and then that's why you see kind of the ATCG, okay, so okay. And then for our the DA project, okay, so we use the DA to first, okay, so figure out the retention time for individual the nucleoside, okay, so ATCG, okay, so that's one example, okay, and then that's retention time, okay, and then we'll run some standard, okay, so figure out the cofactors, okay, eventually that's have to identify, quantify individual the nucleoside kind of amount, okay, and then you can know how many A and how many C and how many T, how many G, okay, so that's kind of area, okay, and then based on kind of some mass, okay, you can calculate kind of the how many ATCG in individual the nucleoside, okay, individual the DNA base, okay. So I'll give you more information probably in the next lectures, okay, and so let me see, that's probably last slide. Okay, that's here the we done the lecture today. Okay, any questions? No. All right, okay. So yeah, next Monday, okay, so we'll have a unit quiz, okay. So yeah, for both the Dr. Yang Ru's kind of units and also the my units, okay. So yeah, you will have 2 quiz, okay. And keep in mind, okay, so yeah, we'll have the quiz in I reserve 1 room, okay, for the quiz, okay. So that's here the 2901, okay, 2901, okay. Please yeah, make sure you come to the right room, okay, right, 2109, okay. Note here the biochemistry lab, okay. So yeah, 2109, okay. And then after the unit quiz, okay, so unit quiz probably take like I will say 1 hour to 40 minutes, okay. So depends on how long of your individual quiz, okay. And after the quiz, okay, so yeah, student in my section, okay, so please yeah, still stay in the same classroom, okay. So I will give out more instruction about here our units, okay, and also I will talk about here the proposals, okay, and also some other informations, okay. And for the student in the Dr. Yang Ru's units, okay, after quiz, okay, you can go upstairs, okay, so to the biochemistry lab, okay, so couldn't remember the number, okay, so probably 2804, okay, or maybe 2814, okay. And then Dr. Yang Ru will give you more information about his the units, okay. Okay. All right, any questions?